I'm Thomas Allen. I'm here from the University of Illinois, and my talk today is going to be on some of our work on proton transfer in particular, and how we kind of are developing some new quantum classical methods to deal with charge transfer reactions. So we're interested in these reactions because they're really a fundamental class of chemical reactions, and they're extremely common. The basic idea is that we have two species which encounter each other. We either have an electron transfer, as in the first equation up here, or we have actual proton transfer, and of course, leads to some kind of charge separation when that reaction happens. And so these reactions, like I said, they happen everywhere. They're ubiquitous in biology. They happen in the electron transport chain. They happen in glycolysis and other biological pathways. Additionally, the idea of transferring protons, especially, or hydrogen atoms or hydride ions, is a major synthetic motif in organic chemistry. A lot of people want to be able to transfer protons from carbon to somewhere else. Nature is very good at that up here, and we are not nearly as good as we would like to be. So one of the things we want to do is understand how these reactions happen, how the environments that they're in influence them, and from that try to develop new synthetic ideas, maybe new materials, especially these days when energy storage, energy generation, charge separation are such big topics. And so one of the problems, in a sense, with this is that proton transfer is a condensed phase process. Now, if it was just like on this last slide where we just had these two species, maybe they're smallish molecules or intermediate-sized molecules, we could do maybe an electronic structure calculation on them, get some information from that, and try and get some insight. But really, what we want to understand is the entire system where we have not just our two interacting molecules, but also the surrounding solvent environment. And the reason that we have a surrounding solvent environment is because this is not really a stable process in the gas phase. So this is kind of a plot of the interaction potential in the gas phase. And so here's kind of a reactant, here's a product. The problem is that the product obviously is not energetically really a product. It just kind of hops back over the barrier. And so the way to get around that is to provide something to stabilize that interaction and bring that side down, which is what the solvent does. So you have some kind of polar solvent interacting via Coulomb potential dipolar interactions, which stabilizes the charge separation and allows this reaction to go forward and to proceed. And so because of that, we're dealing with a condensed phase problem. We have many degrees of freedom. And that's hard by itself, potentially. But the real issue is that we also want to understand the quantum mechanics of what's going on, because what's transferring is an electron or a proton, and these are light species. There's going to be factors like potentially tunneling, zero-point energy, other quantum mechanical interactions. They're maybe important to understanding what's actually happening. And so we need a way to deal with these many degrees of freedom while still addressing the underlying quantum mechanics. And it turns out that the central idea that's important in doing this is separating the whole universe we're considering into an interacting system and environment. Now that idea by itself is not necessarily new. People have been doing it for a long time, going back even to Ehrenfest in the early 1900s. And some of the ideas which exist to either separate these things out or simplify them in some other ways, I've kind of listed here. One of them is QMMM, which some of you may be familiar with, where we kind of also do a quantum classical separation. There's something called surface hopping, which kind of takes some of Ehrenfest's ideas and improves on them. And there also, we can try and sort of build reduced models of what we're studying, make them simpler so that we can solve them exactly, but we give up maybe on some of that detailed description. And the real problem so far with these methods is that they're all making trade-offs that we don't really want to make in our group. The first two can be applied to pretty much any system you're interested in. They can handle an atomistic description. They've been used in problems involving surfaces and materials, biological enzymes, all sorts of things. The potential issue with them, though, is that they introduce ad hoc elements into their description, and those ad hoc elements can potentially lead to uncertainty in your results. If you're in a difficult regime, if you're in something where there's a lot of controversy about what the experiment is actually saying and you're trying to provide some insight, you may spend just as much time arguing about why your approach gets the answer right as you do actually, doing the research itself. Likewise, if we do a reduced representation, the question is, are we missing important features that we are simplifying away, and are some of these irreducible features? So what we want really is a method that works across many different regimes of behavior that can handle all of these, the full systematic description, but also has a very rigorous approach to dealing with the problem. And it's really the important thing that we want to be sure we get right is capturing this full system-bath interaction, the fact that the bath or environment really, the solvent environment interacts with the system, and the system has a corresponding reaction to that. And so what we've developed in our group to address these goals is something called the quantum classical path integral, which I'll kind of briefly introduce here in schematic view. So this, again, is kind of the idea of having all of our system, our universe that we're considering. What we want to do is make a conceptual separation into a system and an environment to start out. So right now we haven't done anything. This is a path integral type expression. We identify whatever relevant degrees of freedom there are to our system, and we kind of treat those preferentially. Then what we do is and I'll show this in just a second, we find this quantity called the reduced density matrix, which means we sort of average or trace across the environmental degrees of freedom, the solvent degrees of freedom, because we don't care about their detailed dynamics necessarily. We're only interested in kind of the average over them and to figure out what the system is doing, in this case, the interacting charge transfer complex. And then the final step, which is what QCPI is really all about, is taking a rigorous kind of classical limit in these environmental degrees of freedom while maintaining the correct interaction between the system and the surrounding environment. And so what that looks like in terms of a few equations, I won't show all of them, but basically the reduced density matrix here is just the trace over here S is the system degrees of freedom, X is the environmental ones. We trace over those. And once you go through and take that classical limit, you do the trace, and that's sort of shown in these papers if you want more detail, is this kind of equation. And what this is basically saying is that our reduced density matrix is equal to some sort of probabilistic average. That's what this P quantity here is. And this thing which we refer to as the quantum influence function, which depends, of course, on the system configuration from these forward and backward propagators, and our initial conditions, and we can calculate it sort of out at whatever time we like, assuming we have enough computational power to do that. This part is pretty straightforward. This is some kind of probability distribution. It can be Boltzmann, it can be a Wigner distribution, just depending on what level of approximation you're dealing with. And that by itself means that we have to do a lot of classical work to calculate the different initial conditions we're averaging over. We also have to do a lot of work to calculate this quantum part, because what this is basically a sum over paths. So we have a forward path and a backward path corresponding to these two propagators, and we sum over all the different possible configurations out to whatever time we're looking at. So this is kind of a schematic example of what we're doing, just one of the kind of snapshots of those. And so what this means basically, well, this is kind of, I'll come back to this in a moment and kind of explain why this is so hard. This is the preview of that. But the important thing, though, is that we've done some work already and shown that this works in spin boson, these reduced cases, where we have other methods to verify them. But what we really want to do, like I said, is we want to look at these atomistic systems. We want to bring in that advantage that QMMM and surface hopping have, where we can look at actual proteins, we can look at actual surface science problems in detail with these. And so the good news about QCPI is that there's no really intrinsic assumptions about the nature of the environment that we make in there. As long as we have some expression for them, we can do classical molecular dynamics on it. And so kind of in moving toward this and kind of showing that we can actually do it, there's a few features that we want. We want a nice, simple molecular dynamics system. We don't want something with too much complexity. You know, we don't want to worry about glassy dynamics and these very complicated, just purely classical effects right now. We do want something that's realistic in terms of its interactions, that it captures this sort of Coulomb interaction that stabilizes our charge transfer complex correctly. And we want the energetics that we get out of this interaction to be correct. And we do, in fact, recover that. I'll talk about that a little bit more in a second. And the other thing that is kind of icing on the cake is that we would like a system where our rigorous approach, where we aren't having these questions of, are we getting this interaction right, that adds something to the picture. And so the system that actually satisfies pretty much all of these is something called the Azou-Bourgi model. And what that looks like in terms of the ND picture is here. It's fairly straightforward. There's a solvent, which is methyl chloride essentially, out here in this box. And then there's also this proton transfer complex here, which is kind of parameterized to represent a phenol-amine transfer from the gas phase. That's actually the curves I showed you before. And so obviously this is simple. It's not a super large system, and it's all kind of diatomics and this sort of triatomic here. It has been used for quite a while, at least in the branch of chemical theory that's interested in studying these proton transfer reactions. It's kind of an underlying model for what's really going on. um, in these kinds of reactions. And the other thing, too, is that a lot of people have studied this, and there is no exact answer for it. It's not something you can solve in closed form. And different people using different methods have gotten different results for, in particular, the rate, which is one of the quantities we're interested in here. And so it's something that we can maybe provide a definite answer for now with this technique. And also kind of demonstrate, since you can see this is an atomistic system, there's no real difference at the computational level of doing this from doing, say, a protein or doing, pick your favorite system. That we can actually handle these kinds of problems computationally. So the challenge to this, again, kind of comes back to this equation. Basically, the problem is that we just need to do a lot of calculations. And the reason for that is we're averaging over some initial probability distribution, so we need to be sure that we sample that sufficiently. And each time we sample that initial probability distribution, that's a different configuration of our system. And for each of those different configurations of our system, we have to sum over all the quantum paths that go into our expression. And so there's a lot of both quantum mechanical and classical work that has to be done. But the good news is it's possible to parallelize these pretty efficiently. And I can show kind of here a scaling curve that basically, as we increase the number of processors that we use, we scale pretty nearly linearly because we can spread this work out very directly. And there's actually some more work we're looking at doing with that too. Another challenge, which we have kind of addressed with help from Blue Water staff and the LAMMPS developers, is that we've got to interface this kind of quantum mechanical forward-backward path summation quantity with a classical mechanical kind of code, which has a certain workflow. And so we kind of need to evaluate the forces and the energies more frequently. We need to evaluate them in a slightly different way. And so we've managed to do that now. We've managed to get that working on Blue Waters and working with the LAMMPS package. And we are also kind of investigating a few more things that we can do. In particular, in talking with the staff, we realized that one of the things we have to do in doing these calculations is we have to move a lot of files around to specify the configuration of our system. And it's possible because Blue Waters has so much memory on each node that we can actually do this in memory instead of writing to disk. And that saves us quite a bit of time on top of the parallelization bonus that we get. And we're also looking at, in fact, spreading out some of this path summation into parallel threads as well, which is promising to potentially make even more use of the resources that we've got here. So, let's see, right, so basically to talk a little bit about where we're going with this, we've, the other challenge involved here is that we have to kind of make sure that we're doing this calculation correctly because there's a lot of convergence parameters involved. So one of the things we've done to sort of check our work, and which we're now kind of wrapping up with, is we have developed a harmonic bath kind of model of this system, which is something we have a lot of experience doing before. We have a lot of other methods to check it. And so we've been working with that to make sure we understand what regime we're in. And that's where we've actually seen, for example, we can measure the rearization energy in the MD and in the harmonic. We see that it comes out to about 0.3 eV, which is kind of in the middle of the range of the standard rearization energies quoted for these kinds of reactions. So we know that our system is realistic. It's still a strong coupling, but for example, right now we're getting this. So this is for the harmonic system. This is pretty much the whole reaction. This is our sort of best converged results right now. And from this, we're doing a few more things to make sure we actually know that we're getting the right answer. But from this, we can pull out the rates. And we're looking at right now, too, what information we can pull out from the classical results of this, what kind of things we can learn from the solvent environment, because we actually are performing an ensemble average as we do this. So we can imagine looking at some statistical quantities which are related to this. And then, so kind of the next step, and the one that we're working on completing kind of over the summer, is to take our fully atomistic system, like I said, that we've got with this LAMMPS code, and that I showed you a few slides ago, and actually running that out also. So this is a comparison of a very short time compared to the last one, but nevertheless some preliminary results from the atomistic regime. And you can see they're actually comparing quite well to the harmonic, and this provides us one of the interesting things, too, we kind of hope to get out of this is because we can treat both the harmonic approximation and then also this fully atomistic system, is to look at how these simplified models actually do compare in a very direct and detailed way with the full description of the system. And so as we go forward and kind of finish converging this and finish running this, that's something that I think we'll be excited to be able to talk about very soon. And kind of in the sense of where we're going in the future with this, okay. In the sense of where we're going in the future with this, we want to obviously finish converging our anharmonic calculations. That's going to take, that's something which right now is mostly just getting everything up and running and spending the time right now. We want, like I said, to kind of investigate what we can get, because unlike some more traditional MD approaches where you may do, and you can obviously do ensemble averaging with that, but we have an actual ensemble here. We have an ensemble additionally across all of the quantum mechanical paths that we're studying. And while it's not immediately clear exactly how that corresponds to kind of a traditional classical thermodynamic average, we're certainly interested in looking at if there are pieces of information we can get from looking at the averages of different quantities over that actual bath and over that solvent system across the different quantum paths. And of course, the next goal, and actually kind of our goal for the fall, once we wrap up this calculation, is to extend this treatment to more complex systems, because sort of this is the proof of concept. Once we've established that we can do it, which we can, we want to start looking at larger, more interesting systems, systems with biological or materials applications. And this is, of course, pretty relevant to me because I've actually received a graduate fellowship from Blue Waters to do some of this work and to extend it to some protein systems. One of the interesting things about this is that although these systems are larger than the one that I've described, I mean, really compared to some of the things that you've probably heard about here for the MD systems, we only really have a handful of atoms. The hard part, of course, is the quantum mechanics. An actual protein system will be quite a bit larger, but it's larger in the classical degrees of freedom, which scale more or less linearly with system size. And it turns out that what makes it hard is how strongly those classical degrees of freedom are coupled to your transferring proton or electron. And interestingly, in bare solvent, that coupling is usually higher. Both experimental measurements and a lot of theoretical justification kind of exists, which point to the fact that in most cases, in protein systems, that coupling is typically lower, which means we do less of the quantum mechanical work in these systems. So we may actually be able to sort of make a very favorable trade here and do those with comparable effort, really, in a lot of ways. Hopefully, it'll depend on where the scaling comes out, but we're really kind of excited to see how that works and how that happens. And so I'll kind of close there and let people ask some questions. But I would like to thank, of course, Blue Waters, the NCSA, and the NSF, both for the time on the computer and also for this graduate fellowship. And of course, my group, which I've shown down here, Nancy McCree and the others. A few of them actually are former group members now, but they've also been helpful. So thank you guys, and I'll take questions now.
About the computational aspect of the code, so you extended LAMMPS?
Yes, so they have, that's one of the reasons we chose LAMMPS actually, is that they're fairly straightforward to extend. So users can write modules which kind of plug in. And what I did was to write basically a module which describes the actual Azeau-Bourget interaction, because it came from the old days of MD where everything was kind of done homebrew, and so not a lot of standard packages incorporate the potential that we needed. And then I also wrote some code to take care of doing those additional force evaluations that we require for our method. So.
Your extensions are what language?
This is all C and C++, pretty much. These days, LAMMPS is written mostly in C++, which is more or less really kind of C with some objects in it. Yeah, we would be interested actually to maybe extend this to other packages. We just don't really have the expertise to do it right now, because we've seen some nice things about the performance, for example, of like GROMACS compared to LAMMPS, but we just don't have any developers right now.
Yeah, second question. You stressed the importance of staying in memory rather than having to go to disk.
Right.
Do you have a measurement of how much memory you're using?
This depends exactly on where the convergence lies, but it's possible actually because we have to store information on the paths, we have to store the description of the system. Basically, we can use nearly all the memory on a node if it comes to that in some cases. We can use a significant fraction of the memory on each node, because what we're really doing is, in order to sort of parallelize it efficiently, we need to kind of have concurrent versions of lamps running within a node. So we have multiple different copies of that running and treating different systems to kind of reduce the communication that goes on. So because of that, we actually use a whole lot of the memory that's on a node, especially if we need if we're in a particularly hard regime where we need a lot of paths basically to converge, we can use 80–90% of the memory on a node. And that amount of memory is not something that's easily available in a lot of other places, which is one of the reasons that Blue Waters has been so good for us.
All right, let's thank the speaker one more time.